they have mentioned that it is the normal human epithelial cells which takes 24 hours to divide. Okay, so as per this, the biggest stage is this G1 cycle. Then there is S phase, and then there is G2 phase, and then there is M phase. So the sequence of events is G1, S, G2, M. Okay, G0 is a separate thing. G0 is a separate thing. So it is whatever the diagram which they have mentioned over here is in context to the 24 hours division or 24-hour division of the normal human somatic cell, okay, which it takes to divide. And on that basis, this has been explained. So the biggest phase is G1 phase, then there will be S, then there will be G2, and then there will be smallest is the M phase. G0 is a separate story. It is not considered as such as a part of cell cycle. So G1 time duration will be 10 to 11 hours. S will be 8 to 9 hours. G2 will be 3 to 4 hours, and M phase will be 1 to 2 hours. Okay, this is how the time has been divided. For different cells, this can be different. Like yeast can complete the cell cycle in 90 minutes. E. coli, it will be 20 minutes cell division time. Okay, so you should know this is as per the human somatic cell. So the biggest phase is G1. So the biggest phase is G1. So as such, we can divide cell cycle into two phases, that is interphase and M phase. So interphase is when the cell is not dividing, then you can classify it into all of these phases where G1, S, G2, and then we have M. Now M phase, again we have karyokinesis or nuclear division, or we have cytoplasmic division. Okay, so you can divide them into karyokinesis as well as the cytokinesis. So first nucleus will divide and then cytoplasm will divide, as you know. So the exact cell division, that is M phase, it only lasts for an hour. Okay, it only lasts for an hour. So the interphase is more than 95% of the duration of the cell cycle. Then corresponding to the rotochromosome, as I mentioned, the nuclear division is referred as karyokinesis. Cytokinesis is what we call as the cytoplasmic division. So karyokinesis, we have normally prophase, metaphase, anaphase, telophase. Over here, see, M stands for normally mitosis, but it can also be meiosis. Okay, it can also be meiosis. Keep that in mind. Because the cell can undergo meiosis also, if it's a gamete producing cell or if it's a gamete mother cell. So we're discussing about this stage where it is the G1 is referred as the gap 1 or growth 1 phase. S is referred as the phase which comes after G1, and then we have gap 2 or growth 2 phase. Okay, both the terms can be used. Gap and growth both can be used. So see, basically in the G1 phase, there is no replication. Notes just try to understand. Maria stylus સરખું ચાલતું નથી, there was I would have drawn and explained. તો હું generally તો આ circle આખું draw કરીને સમજાવું છું, but unfortunately stylus is not working, so I have to explain verbally. So during G1 phase, the cell is metabolically active. It will continuously grow, but there will be no DNA replication. Especially. So the cell will form ATP, it will grow faster. Okay, cytoplasm will increase, but there is no DNA replication. DNA replication occurs in S phase. DNA replication occurs in S phase, one of the very important questions. So now they mention two things, that if it is the 2C content, it will become 4C. And if it is 1C, it will become 2C. So here as such, C stands for chromatin. We have covered nucleus, so you know what is chromatin. Here usually C stands for chromatin. But in your textbook and sometimes in question, it is also referred as chromatin. It is also referred as chromatid. So what they're trying to say is that there is doubling of the genetic material, or there is duplication of the genetic material. The genetic content doubles. Okay, so now over here, keep one thing in mind, that whenever we refer to 2N, the diploid cell, it is 2C. So if it is 2C in G1, then it will become 4C in okay, in the after the S phase. So it will become after the S phase, it will be 4C. If it is haploid cell, it will be what we refer as 1C. Haploid cell no content je hase, that will be 1C. But after the S phase, it will become 2C. Okay, people get confused, so kindly pay your attention for this. Now see what it means that usually we mention that the chromosome number remains the same. Okay, chromosome number remains the same. But chromatin or chromatid, but what we call as chromatin, or in higher books it is referred as chromatin only, it will double. It will double in the S phase. Now what does it mean? It is like, let's say if I have 100 rupees, then I'll be able to give 50 rupees to two other people. Okay. But if I have 50 rupees, then I won't be able to give 50 rupees to two people. Okay. So it is same. The genetic content will double, but we do not say that the number of chromosome will increase. So the number of chromosome remains the same. As you can see over here, the number of chromosome remains the same. Okay, so let's say if we talk about human cell. Okay, let's say if we talk about the human cell. Okay, so see, human cell, diploid cell has how many chromosomes? કેટલા chromosomes है diploid cell में humans ना?
46.
46. So it is in the 2n condition. Okay, my stylus is not working fine, so I have to write with my fingers anyways. So my handwriting will not be that good. So it is 2n equal to 46. So this is what we refer as 2C beta. This is what we refer as 2C. So after S phase, it will become 4C. So as such, the genetic content will be of 92 chromosomes. The real genetic content 92 chromosomes nu thase. But we will not use that term that the chromosome number had become 92. તમને question પૂછે તો chromosome number remains the same. Why it is so? Because after the G2 and M phase, it will form two cells. No, then two cells, if it is formed by mitosis, it will have 46 chromosomes each. No, so અહીંયા આટલું કન્ટેન્ટ હશે 92 chromosomes નું S phase પછી તો જ આ બે બનશે ને, બાકી તો પોસિબલ જ નથી ને બેટા. So that's why, but we never say that the chromosome number will double. We always refer that the chromosome number remains the same while the genetic content has doubled. So did you get this part, beta?
Yes, sir.
It is important, right? It is very important. A lot of people mess it up. So kindly understand. If it is a haploid cell, then it will be 1C. Then after S phase, it will be 2C. Got it? So if it's, let's say, haploid has 8 chromosomes, it will be 16. The content genetic material will be of 16 chromosomes, but it doesn't mean that but we will never use that term that the chromosome number is double. But we have to understand it in that manner. Okay, it's the same example. If I have 100 rupees, I can give 50 to 2 persons each. So એટલું content થઈ જશે, પણ આપણે એવું કહેશું નહીં કે double થઈ જાય. Okay, then in S phase, the other events will be centriole will duplicate. Okay, then we'll go to the G2 phase. But before going to G2 phase, there is a checkpoint. There is a checkpoint over here. Oh, sorry, there's a checkpoint over here between G1 and S. Okay, so it will check whether all the events which are supposed to occur in the G1 phase had been properly formed or not. Then it will go to the G2 phase. And then after G2 phase, there is also a checkpoint over here between G2 and S. So there are two checkpoints. First is G1 and S, another is between G2 and S. Okay, they also ask these questions about the checkpoints, so you should know. Okay, now what these checkpoints will do is just like a check post. When you enter in a city, there is normally a police checkpoint outside the city which checks whether there is anything illegally which has been carried in the vehicle. So similarly, this is also a Similarly, this is also a checkpoint which will ensure that whether the events in the G1 phase had properly occurred over here, and to check કે અહીંયા S phase માં બધું proper થયું છે કે નહીં, because then there will be M phase. તો M phase માં તો cell divide થશે. And if there is any mistake in DNA replication or chromosomal content, then two daughter cells will not be normal. So that's why there will be checkpoint between G1 and S and between G2 and M. So during G2 phase, there is RNA protein synthesis, which is required for mitosis. Okay. Cell growth will also continue. And then it will divide in M phase. So you should know that the checkpoint is first checkpoint is between G1 and S, and second checkpoint is between G2 and M. Okay, so these are what we have, two checkpoints. It is mediated by cyclins or cyclin-dependent kinases If this checkpoint will not function, then cell may divide uncontrollably. It can lead to cancer. It can lead to formation of abnormal daughter cells. Okay, it can lead to unequal distribution in the two daughter cells of the genetic content. So all of these things will be checked at this checkpoint by these cyclins, and the cyclin and CDK are mainly proteins. They are mainly proteins which will check this. Okay, now over here we are talking about G0 phase. So see, as such, G0 phase is also termed as the quiescent phase. It is also termed as the quiescent or inactive phase. So if the cell is supposed to enter G0, it will enter from G1. And if cell is supposed to divide, then it will go into S, G2 and M, which we had discussed. એટલે જો સેલ જે છે ડિવાઈડ નહીં થાય, ઓકે, તો શું થશે કે G1 માં જતો રહેશે ડાયરેક્ટ. Now G1 is metabolically active stage, only division will not occur. So it is usually seen in neurons and heart cells, where the cells will never divide in their lifetime. As you know, neurons will never divide. The number of neurons are fixed. Okay, their number may decrease as we grow old, but their number will not increase because they cannot divide. So some cells which doesn't have a capacity of cell division, they will undergo G0 phase. Some of the heart cells can divide if needed, especially in cases of the injury or cell death. Okay, sometimes they may divide. They are metabolically active. Okay, and some of them have capacity to divide, else they will not divide and they will be in that stage. They are differentiated cells. Okay, so neurons or heart cells are examples of the G0, also referred as the inactive or quiescent state. And if cell is supposed to go in G0, it will directly go from G1. That is also a question. Okay, so now Rahi, answer me, what will be in this case over here? They have mentioned in this box. अच्छा, box માં જે question છે એમાં શું answer આવે બેટા? This thing. This has also been asked, okay, in exam.
Sir, in G1 phase 16 chromosome and
Okay.
S phase 32 chromosome.
No, beta, we can never, as I told you, we never use that term. Chromosome number remains the same. Only genetic content doubles. So after S phase also it will be 16 chromosome. Okay, so what they've asked you is, they've asked you about the DNA content also. Okay, after M phase, the number of chromosome will be again 16. Okay, now what about the DNA content? So that is 4CNO. So if in the G1 phase it was 2C, okay, S phase 4C, after M phase again 2C. Okay, so in comparison to the G1 phase, if it was 2C, then after S phase it was 4C, so it will double. So the DNA content will be double. Okay, See, that is what they ask. What is it? The G. So in the G2 also it is 4C. G2 also S and G2 it will be 4C. Chromosome number double ना करती खोटू पड़ सक. Chromosome number always remains the same, but the genetic content doubles. खबर पड़ी क्या नहीं बेटा?
Yes, sir.
Now we'll talk about the mitosis. So mitosis is comparatively easy. Meiosis is slightly difficult to understand. So in mitosis, from single diploid cell, two diploid cells are formed. So if it is 46 chromosomes over here, we will have two daughter cells which will also have 46 chromosomes, which will also have 46 chromosomes. Okay. So it is divided into four phases, that is telophase, prophase, metaphase, anaphase, as you know. Okay. Now we all know that it is a part of the karyokinesis or nuclear division. So it will the first phase after S and G2 phase. It will be first phase after S and G2 phase. Okay. Now it is marked by initiation of the chromosomal condensation. Okay. So usually the chromatin will start to get condensed or will start to get transformed into thread-like structure called chromosomes. And this process is what we refer as chromatin condensation. Okay, so the chromosomal material will start to get untangled. Then the centrosomes will also duplicate. Centrosome, because as you know, centrosome contains two centrioles. So these two centrioles will also go to the opposite ends of the cell. It will also go to the opposite ends of this, will start to move towards the poles of the cell. Okay, so this is the chromatids can be seen, chromosomes will be formed. So the centrosome will undergo duplication. Okay, it will move towards opposite poles as I have explained. And then it will form what? It will form the spindle fibers. So from this, there is formation of the spindle fibers. Spindle fibers will be formed like this from the centriole, as you know. So the esters are what we the centrosome has micro. This spindle fibers are termed as microtubules. Spindle fiber are basically termed as microtubules. So in animal cell, we call this sort of spindle fiber formation as amphiestral. Why? Because two centrioles are arranged at two different poles of the cell. And they are made up of the protein, which is tubulin. So tubulin is, microtubules are made of protein tubulin. Now when you view it under microscope, the cell organelle will disappear. Cell organelle will disappear. So you can see over here the chromosomes has started to form. Okay, you can see over here that the chromosomes have started to form. Okay, over here. Then the nuclear envelope has been had broken down. Nuclear envelope is disintegrated. Now you can see the chromosomes. So it is in the late prophase, and then all the organelles will disappear. Now in mitosis, in metaphase, what will happen? The chromosomes will start to get arranged at the equatorial region. So this is the transition to the metaphase. There's also onion root tip experiment in your syllabus. So as you can see, chromosomes have arranged at the equatorial region of the cells. You can see over here there is centrioles, there is spindle fibers which has been formed, and chromosomes are arranged at the equatorial region of the cell, which we refer as metaphase plate. We refer as metaplate phase. This is the spindle fibers. Now in the case of the anaphase, chromosomes will split from the centromere. Centromere, I hope you know, where two chromosomes are combined. So they will split in from the centromere. Okay, so as you can see, the chromosomes had split from the centromere. Okay, centromere thi banne chromatids alag thase. And then these chromatids will move towards the opposite pole. From the center, they will move towards the opposite poles. And then finally, from this split chromatids, we will have nucleus formation, and then from this there will be formation of the two different cells, which will be 2n only. As you can see, the nuclear envelope will again form, organelles will appear. So it is just like interphase nucleus. It's just like a interphase nucleus. So from this, the important sentences, okay, are, as you know, this is the thing they ask, that this spindle fibers are connected to kinetochore. Kinetochore is a disc-shaped structure on the sides of the centromere. Okay, so you should know how it is characterized. Okay, the alignment is referred as metaphase plate. If you have any doubts, you can ask me. Okay, I'm just explaining whatever is important from my point of view. Okay, then there is this chromosomes will be future nucleus, because these two cells which will have transformation of this split this chromatids into their nuclei. So they'll be directed towards pole. Telophase, the chromosomes will lose their individuality, opposite of the prophase. Telophase is opposite of the prophase. Whatever events had occurred in the prophase, those events in the telophase will be opposite to that. So it is termed as opposite to prophase. Okay, nuclear envelope will develop, these all organelles will reform. So karyokinesis will end with telophase. Okay, so now if you have any problems, you are free to ask from this.
No, sir.
Now see, cytokinesis, it will be separate in animal cell and in plant cell. So over here, if you see, they have shown you the animal cell cytokinesis. Okay, so now I explain how it is. So see, if you see this furrow, okay, the cleavage like structure, a furrow which you see, okay, this starts from the periphery. This furrow or cleavage starts from the periphery, and it will go into center. This will go into center, where this will meet, and then there will be formation of two separate cells. The animal cell માં શું થશે કે જે furrow કે cleavage બનશે, it will start from the periphery, it will go into the center. Okay, so in animal cell it is achieved by the appearance of furrow, and it will go into the center. It will deepen and it will go into the center, and then it will split the cytoplasm into two. So this is the cytokinesis in animals. Now plant cell, it is different. Okay, plant cell, the process is different. Okay, so let me explain that. So let's say plant cell have a cell wall, as you know, and they do not have centriole. Okay, they do not have as such centriole. So they have a nucleus in the center. So what will happen now in plant cell? The division will start from the center. In plant cell, the division will start from the center. So over here, there is a cell plate like structure will be formed. Okay, and then it will split plant cell into two unequal cells. It will split plant cells into two unequal cells. And such a structure is referred as what we call as cell plate. This is what we call as cell plate. Now, what will form cell plate? So in plant cells, the cell plate is formed by dictyosome. Dictyosome is another name of Golgi body. In plants, we refer Golgi as dictyosome, endoplasmic reticulum, and spindle fiber. So it is opposite to animal cell. Okay, plant cell માં શું થશે? It will be center માંથી start થશે. Okay, this is what they have mentioned. Okay, because plant cell have a cell wall, so it will start from the center and it will go towards the periphery, Okay, animal cell માં ઊંધું હતું, periphery માંથી center. And this cell plate is formed by these three: dictyosome, ER, and spindle fiber. The cell plate is formed by dictyosome, endoplasmic reticulum, and spindle fibers. Not given in textbook, but the question had been asked. And then it will split plant cells into two unequal cells. Animal cell માં normally be equal shape na cells બનશે. Plants ના case માં એવું નહીં થાય. The only reason is they have cell wall. The only reason is because they have cell wall. Okay.
Okay, sir.
So this is Dictyosome is Golgi body. The name of is used in relation to plant cells. So remember that in relation to. So significance you can directly read, okay, for normal growth and development. And then if you have any issues, you can ask me. Okay. Usually you should know one thing, that it maintains N/C ratio. So each cell has particular size of nucleus in relation to its cytoplasm. If it gets disturbed, and it should get disturbed as the cell will grow, then only cell division can occur. So once the cell division can occur, it is again restored to the normal level. Okay, તો દરેક સેલમાં એક ન્યુક્લિયસની સાઇઝ અને સાઇટોપ્લાઝમનું એક રિલેશન હોય, મતલબ કે અમુક સેલની સાઇઝ પ્રમાણે એ સાઇઝનું ન્યુક્લિયસ હોવું જોઈએ. So if that is disturbed, then the cell will undergo division. And a disturb થાય તો જ division મળે. Cancerous cells માં this control is lost. Okay, so then it will not divide, or they will not die when they are supposed to die. So that is not happening in normal cells. So the role of the metastasis is usually to restore. Now we have meiosis. Okay. So meiosis is very important. Okay. So meiosis has two division. That is, it has M1 and it has M2. So once it starts with the 2n cell, the first division will form the half the number of chromosomes, and second is like mitosis. So you will have 4 haploid cells from single diploid cell, as you can see. Okay, it is needed for the reproduction of the gametes in sexually reproducing organisms. Okay, so this you can read basically what happens over. It is important for questions. So now I'll just explain what will happen in the meiosis. These are the phases. If you understand prophase 1, everything is very easy. So prophase 1 has different phases, like it has lactotine, then it has zygotine, then it has pachytene, then it has diplotene, and then it has dikinesis. Okay, so see, I'll explain what happens in the lactot, that is prophase 1. Okay, so the prophase 1 is the first phase of meiosis 1, and it has 5 different stages. So see, first let me explain by 2 different chromosomes. So in the prophase 1, in the lactotine, okay, you can see the chromosomes will gradually become visible. Okay, I'm showing you by 2 different chromosomes. Now in the zygotine, let's say this is 1 chromosome. Okay, and then we have, just a moment, this is another chromosome. Okay, this is another chromosome. So these chromosomes will be arranged upon one another. They will arrange upon one another, and this is what we refer as synapses. So humans ma 46 chromosomes હશે ને તો 23 આવી pairs બનશે. There is pairing between these chromosomes which we refer as synapses. And such chromosomes are termed as homologous chromosomes. Such chromosomes are termed as homologous chromosomes. The compaction happens in lap 13. Now the synapses occurs in this phase. Now what are homologous chromosomes? Also it is needed in genetics. Okay, so as we know, we all receive one set of chromosomes from mother, one set of chromosomes from father. So 23 comes from mother, 23 comes from father. So these 1 to 23 from father, 1 to 23 from mother. So these are referred as homologous. These are 1, 1, 2, 2, 3, 3. So these are all referred as homologous. Got it? This is how diploid organism is formed. So such way there is pairing which will happen. And this is what we refer as synapses. This is what we refer as synapses. So such complexes are referred as synaptonemal complex. Such complexes are referred as synaptonemal complex. So you can see there is multiple pairing, multiple synapses are there, and they form multiple synaptonemal complexes. Now we refer to this as a bivalent or tetrad. Why? What it means? Why tetrad? Because normally a chromosome will have two chromatids, but if you look over here, you can see two chromatids of this blue one and two chromatids of this red one. So it appears as if it has four chromatids. Ideally બંનેને હશે બે બે ક્રોમેટિડ જ, પણ એકબીજા પર એવી રીતના સિનેપ્સિસમાં અરેન્જ થયેલા હશે કે ઓવરલેપિંગ સ્ટ્રક્ચર હશે. So it will look like there are four chromatids. It will look like there are four chromatids. So it is referred as tetrad or bivalent. So it is referred as tetrad or bivalent. Okay. So keep that in mind. Now, ક્યાંય problem હોય તો then ask me. બાકી આગળ પછી ખ્યાલ નહીં આવે. Now the third phase is packeting. Now the third phase is packeting. Packeting is highly important. So now what will happen? That this bivalent will appear as tetrads. Okay. And at certain regions, these chromosomes will come in contact. These homologous chromosomes will come in contact at certain. Let's say over here they are coming in contact. So such regions are termed as recombination nodules, where the crossing over will occur. Okay, between two non-sister chromatids. Now what it means, crossing over એટલે genetic material નું exchange થશે આ red and blue વચ્ચે. So red chromatid and blue chromatid are of two different chromosome. So they are termed as non-sister chromatid. They are termed as non-sister. If it is of single chromosome, we call it, these are sister chromatids. But as red and blue are different chromosomes, okay, they are referred as non-sister chromatids. Keep that in mind. So now there is genetics. So the place where they will come in contact. So the place where these chromosomes will come in contact. Okay, there will be, let's say there will be exchange between them. So at certain regions there is exchange of genetic material. Let's say over here there is exchange between of genetic material, and this has been referred as crossing over. Now crossing over is mediated by enzyme called recombinase. It is mediated by enzyme called recombinase. So what will happen? See, this is the reason. Crossing over is the reason why we have certain characters from mother, certain characters from father. Okay, we are not identical copy of any one of them. Yeah, it may happen that we may have more characters from mother or we may have more characters from father. So it leads to recombination of two chromosomes. It is completed by the end of pachytene. Okay, and the chromosomes are linked at the sites of crossing over. So still this is in tetrad form, and still they are attached where the crossing over had occurred, like this. Okay, so crossing over is nothing but the places where chromosomes will come in physical contact with each other, owing to synapses or owing to synaptonemal complex. Recombinase will function. There is exchange of genetic material. Now in the diplotin there is dissolution of this complex, and the bivalent will, or tetrad is, still see there are tetrads. They start to separate. So this tetrad will start to separate, but still they are in contact like this. Okay, so let me just show you with two different colors so it will be easier. So this blue and red which we talked about. So now what will happen that mainly this is, let's say, blue, and this is, let's say, red one. So tetrad is still there, but still they start to go away like this. Okay, so the places where they are in contact, so the places where they are in contact, like see this region and this region, it is a X-shaped structure, no? And this X-shaped structure is what we refer as, or the chiasmata, is what we refer as the chiasmata. They are going far away from each other, but still they are attached at the sites of crossing over. That is what we refer as chiasmata. Now there is this sentence that in oocytes of some vertebrates, diplotin can last for months or years. So if you know about human reproductive system in the female of the human, okay, the oocytes will be stopped in the diplotin stage after birth. And when the first ovulation will occur, or when the first menstrual cycle will begin, this will complete its M1. So this comes in the 12th in the chapter of human reproduction in female gametogenesis. So the female ovulation will begin at puberty, and prior to that, from childhood to the puberty, these oocytes are stopped in the diplotin stage. And when the puberty will start, this will also start to mature. M1 will be over, and then that is meiosis 1 will over, and the ovulation will begin during the first menstruation. That is what we refer as the menarche. There is also a question you should know is in diplotin of the prophase 1. Now next is in the diakinesis, it shows the end of or terminalization of chiasmata. It shows the end or terminalization of the chiasmata. Okay, so the chromosomes are condensed and there is a meiotic spindle is formed. Okay, so there is meiotic spindle will be formed, nucleus will disappear, envelope also breaks down, and it will go to the met. So they also ask this question that which stage shows terminalization of chiasmata? Answer is diakinesis or diakinesis, as you can pronounce as you want. So this is what we have in prophase 1. This is what we have in prophase 1. તો આમાં કઈ ડાઉટ હોય તો બોલ બેટા, બાકી કેમ કે માયોસિસ માં પ્રોફેઝ 1 જ મેઇન છે, એ આવડે તો બધું આવડે.
No, sir.
Okay. Now see what will happen. Still they are bivalent, huh? Still they are bivalent. So see, this is what prophase 1, you can see this sort of structure. So they have shown two chromosomes over here. These are nodules. These dots which you see are recombination nodules. Now see in metaphase, still they are in as such bivalent. They are very near. Atlo gap nahi hoya. અહીંયા જે દેખાય છે ને એટલો gap nahi hoya. They are very near. Okay, ideally this is one chromosome, then this is another one, so they are very near. Still they look as if they are very near to one another. Okay, so still you can see bivalent over here. Okay, but they are arranged on the what we call as the equatorial plate. Now in anaphase, as you can see, now the anaphase 1, the bivalent will separate, tetrad will separate. So there were two chromosomes, so 2 will go on this side. After recombination, 2 will go on this side. Remember, sister chromatids will not separate. Remember, sister chromatids will not separate. So finally, at the end of the M1, you will have two chromosomes each. Okay, so this will be 2 chromosomes each, and these are N. So if we have started from the 2N cell, we'll have 2 haploid chromosomes at the end of this, as you can see over here. And you can see this different color, light red and dark red and light blue and dark blue. And this is why, because there was a recombination. So અમુક homologous content mother નું આવ્યું હશે, અમુક father નું આવ્યું હશે, એ આપણામાં genes તો હશે ને એટલા માટે. Okay, that's why you can see this different color codes, light and dark. That shows recombination which had occurred because of the crossing over. Because of the crossing over. Okay.
Okay, sir.
So now in these homologous will separate, but sister chromatids are associated. Also a question. And finally, we'll have two cells at the end of the telophase, which are haploid. So also sometimes interkinesis is between two meiotic division, which we call as the short-lived. So there is some dispersion of chromosomes, is the pause or a gap between two stages. Now, meiosis II is just like, it's very easy. It is just like mitosis. So see over here, we have, અહીંયા જો બેટા બે cells તો બની ગયા હતા ને haploid, તો આગળ આપણે વાત કરીએ કે નહીં. Okay, you can see over here. See over here, you can see there are two chromosomes each in the meiosis I. Now it is just like mitosis. Now it is just like mitosis. So see, you can see it is arranged at the, in this is where in the prophase 2 you can see chromosomes over here. Now they'll be arranged at the metaphase plate. Same, બંનેમાં same જ થશે. They'll arrange at the metaphase plate. This is the metaphase. Then in anaphase, what will happen? Centromere will split, so chromatids are getting separated, going towards the pore. Okay, so now અહીંયા centromere split થશે અને homologous chromosomes of the sister chromatids will split and they will go away from each other. And then finally you have two cells. These are again haploid. Same over here, you can see sister chromatid splitting and you have 4 haploid cells from single diploid cell. You have 4 haploid cells from single diploid cells. And as you can see, it depends on the gametes. Like if in the male, some will have, 2 may have X chromosomes, few may have Y chromosomes. Female all will have X chromosome, and these different color codes, light blue, light red, and dark red, dark blue and light blue. This represents recombination that occurred in the crossing over. So finally, we'll have 4 haploid daughter cells. Significance is evolution to maintain chromosome number and to produce variations. So this is what we have beta in the meiosis. તને કઈ problem હોય આમાં તો બોલ.
No, sir.
In this chapter, cell cycle, prophase 1 of meiosis 1 are most important for question. Okay, so kindly read these and then go through it. તારે next શું કરવાનું છે, કયો ચેપ્ટર કરવાનું છે બેટા?
Structural organization.